Hi again. This is the second part of the second half of a tutorial to help you continue to write your lab. Okay, so what we're going to do now is we're going to talk about something called transformation efficiency. Transformation efficiency is a way to calculate how good you were at getting your wild type cells to take up exogenous DNA, become transformed. They become what's called a transformant, which is a transformed cell. How good were you at that? So you can use that information, that quantity, that value, to support your claims in the first part of your discussion section when you're just exploring your data in general, when you're exploring the claims that you made in your introduction, using your data as evidence to support those claims. But then you can also use this transformation efficiency value as a quantitative way to talk about any variability in your data, otherwise known as your error analysis, because the second and third actually parts of your discussion involve your error analysis, and then the last part is looking at talking about improvements, which are ways to improve or essentially reduce the error. So let's talk about lesson 4, because you need to be able to complete lesson 4 to calculate transformation efficiency, and then I'll briefly talk at the end about how you can use it. So lesson 4 is right in your lab packet again. It's on page, 47 in your lab packet, right here, page 47. And your lab packet does a really good job of simply walking you through the calculation. This is the formula that we're going to use to calculate transformation efficiency. We need two basic values. The first number is the total number of colonies growing on a particular treatment. And the second number is the amount of DNA that you put on that particular treatment. So it's the number of cells that actually grew. And because we grew those on a selective media, we know that all those cells are transformed. So we're looking at transformed cells divided by the total amount of DNA that was included in that treatment. So before we talk about the way to calculate the transformation efficiency, let me just briefly say, why do we care? And the reason we care, in the classroom, we care, it's sort of bragging rights. It's who was the most awesome. All of you are awesome, but some of you will get better results, better transformation results than others. But if you can imagine, in a biotechnology lab, scientists would want to maximize the efficiency of their protocols. So your protocol was maximized for the particular organism that you're using and the particular plasmid that you're using. So that's why we, for example, were in the water bath at precisely 42 degrees for precisely 50 seconds. So all of those things are to maximize transformation efficiency. Okay, so like I said, we're going to use this transformation efficiency equation right here. So let's look at how we get this numerator and this denominator. So that's what they're telling us here. So number 1 is getting the numerator, the total number of green fluorescent colonies growing on this particular treatment. And number 2 is the total amount of DNA that you added to that particular treatment. Now, like I said, let's talk about number 1 first, and let me just add that this particular procedure walks you through how to calculate your transformation efficiency on your glowing cells. So you were talking about your LB and ampicillin and arabinose treatment, but you could also calculate transformation efficiency for your treatment that was plus P-glow, but that just had the lauria broth and the ampicillin with no agarose in it. No arabinose, pardon me. So it's important to note that your transformation efficiency can be calculated with either one of those plus P-glow treatments. So this number, the numerator, is really easy to get because it's just determining the total number of cells on that treatment, and you just counted them. So let's say, for example, that on my plus P-glow amp era plate, that's the plate that I'm going to talk about right now, I had 10 colonies. I just put that number in right here. That's all that is. So I'm going to put the number 10 in right there. And the way that this lab packet is written, we're just going to put our numbers in their graphic organizer, and then we're going to calculate the number in the end. So put your number, whatever your number of colonies is, put it in right there. I'm going to say my number is the number 10. Number 2, remember, is we're calculating the denominator of that transformation efficiency equation. So we have to figure out how much P-glow we put on that particular plate. In this particular case, we're looking at the LB amp era plate. So it requires 2 different pieces of information. The first thing we have to do is figure out how much total P-glow plasmid you had in each lab group. Not total for the class, but just total for your lab group. So how do we figure that out? Well, we're actually going to get the numbers right from your procedure. Here's the total DNA equals the concentration of the DNA times the volume that you put in the microcentrifuge tube. The concentration of the DNA, this is a little more confusing than just counting the colonies because you weren't responsible for some of these numbers, but they're contained right in this procedure. So let's just see if we can pull them out. The concentration of the DNA is right here. It tells you it's 0.08 micrograms per microliter. So it's this number right here. And all that means is that in that clear glass vial that had the P-glow in it that we passed around and everybody took some of for each of your investigations, I mixed transformation solution, a specified volume of transformation solution with lyophilized plasmid. And all that did was create a solution. Lyophilized means freeze-dried. All it did was create a solution of DNA. So the reason you don't know anything about this is because I did it for you. So I just rehydrated the freeze-dried DNA. And so you're going to put the number 0.08 in right here. And then you're going to multiply that times the volume of DNA that you used. Now W2, if you recall, you used the yellow inoculation loops to move a film of DNA to your plus P-glow microcentrifuge tube. Most of you used the green microcentrifuge tube to show plus. Some of you used red. So we're going to estimate that loop, that film on your loop was 10 microliters. In W3 and W4, we changed the procedure slightly, and we actually used the micropipetters, adjusted them to 10 microliters. So everyone's going to use the same value. So that's the volume of DNA, the total volume of DNA that you used in your plus P-glow treatments. So all you're going to do is multiply 0.08 times 10, and that just equals 0.8. And that number is going to go in right here. So that's all that's happening now. Now that total amount, all we have to do now is we have to multiply that total amount by the fraction that you put on each treatment. So that's the number that we need to get now. So let's go to page 49 and let's figure out how we're going to get that fractional amount. How much did you put on each individual plate? Okay, so here is how we calculate the fraction. You need to have the total volume that you spread on each plate. This is actually an error here. If you look at the formula, this is showing you the LB amp plate, but it really means the LB amp era plate. The era should be right here. If you were calculating the volume to put on your LB amp plate, which you could also do, like I said in the beginning, then this would be correct. And then over the total volume in the microcentrifuge tube. So you're just getting the fraction. Now, this is right from your procedure. You spread, if you remember, you used the disposable pipetters to pipette 100 microliters of cells that were mixed with the P-glow. And remember, you put them a little bit of, you had 250 microliters of transformation solution. You had 250 microliters of Loria broth. And then you had 10 microliters of DNA. And so that's where the 510 comes from. That's your total sample in your microcentrifuge tube. So you had 100 microliters, that number's right there, over 510 microliters. That's right from your procedure. And that gives you the fraction of DNA. So I'm going to let you do that math yourself. I'm not going to tell you the answer. So now you have the fraction and you have the total. And so you're just going to multiply those two numbers together. And that's what you see down here. to get the p-glow spread on a particular treatment, you have the total, that number was up here, this number, times the fraction, that number is right here, and you're going to enter that number here, that number right there. And that number is going to tell you how much DNA is spread on each treatment. So now you know for each treatment how much it is, or each of your treatments with plus p-glow. It's slightly different for your ones without p-glow because the total volume is a little different. But you're not going to calculate transformation efficiency when you haven't added a plasmid, because that doesn't make any sense. Okay, so now that you know that, that's your this number right here is your denominator. So if you remember from our original equation, we need the numerator. That was just the total number of cells. In my case, for the example that I used, I was going to put the number 10 in here. And then the number I just calculated, I'm going to put in the denominator. And that's going to give me my transformation efficiency. And that's this number right here. Transformation efficiency, the units is transformants per microgram. A transformant is just a transformed cell per microgram of DNA. That's going to give me this value. Now, these numbers are going to be really small. And because they're going to be really small, I'm going to ask that you express them in scientific notation. And so this is just a couple questions that get at making sure you understand how to convert your numbers to scientific notation. And then I'm asking you to convert your calculated transformation efficiency into scientific notation. Now, the question is, where can you use this number? Well, if you refer now to your yellow sheet that I gave you, the scoring sheet that I gave you, take a look at that and you'll see it gives you your expected transformation efficiency for this lab. So you can do two things with this particular calculation. You can use it in your discussion section to provide evidence for transformed cells. You can also use it in your error analysis to talk about your percent error. Okay, that's how you calculate transformation efficiency. There will be another lab work session tomorrow. If you have any questions, feel free to send me an email, and enjoy the rest of the snow day.